Hi friends, my name is Nandita.
Good morning dear students. Today we are going to discuss about locomotion and movement from chapter 20 biology class 11 CBSE syllabus. Amoeboid movement. Amoeboid movement is the most typical mode of locomotion in adherent eukaryotic cells. Amoeboid movement is a crawling or oozing motion that some cells and unicellular organisms use to move. It characterized as by the following. Pseudopodia. The cells form pseudopodia or false feet, which are convex extensions of the cellular surface. Cytoplasmic flow. The cells uses cytoplasmic flow to pull itself forward by changing the thickness of cytoplasm in different areas of the cell. Plasmocell and plasmogel. Cytoplasmic fluid turns into plasmocell, a thinner, more watery version of cytoplasm which flows forward and pulls the membrane with it. At the tail end, cytoplasm thickens into plasmogel which forms a wall to prevent the plasmocell from flowing backward. Locomotion. Amoeboid movement can be used for walking, gliding, and swimming. Amoeboid movement is used by a variety of cells including white blood cells, smile molds, and protozoals. Children, let us see the amoeboid movement like crawling. Crawling is one form of amoeboid movement which starts when an extension of the moving cell pseudopodia bends tightly to the surface. The main bulk of the cell pulls itself toward the bound patch. By repeating this process, the cell can move until the first bound patch is at the very end of the cell, at which point it detaches. The speed at which cells crawl can vary greatly, but generally crawling is faster than swimming, but slower than gliding on the smooth surface. However, crawling does not become notably slower on uneven and irregular surfaces, while gliding becomes much slower under such conditions. It seems that crawling can be either left driven or actin driven, means see sections below, depending on the nature of the surface. Children, now let us discuss gliding. Gliding is similar to crawling, but it is characterized by much less adherence of the surface, making it faster on smoother surfaces, which requires less traction but slower on more difficult and complicated surfaces. Some cells glide with the same mechanism as crawling but with larger pseudopodia and less surfaces adherence. Other cells use a different method to glide. A small patch of the cell already touching the surface binds to the surface, after which the cytoskeleton pushes or pulls on the anchored patch to slide the cell forward. This difference forms the aforementioned mechanism in that the cells does not extend a pseudopod, so there is relatively little deformation of the cells as it progresses. Now let us discuss about swimming. Many different prokaryotic and eukaryotic cells can swim, and many of them have either flagella or cilia for that purpose. These dedicated structures are not necessary for swimming through, as they are amoeba and other eukaryotic cells which lack flagella and cilia but can still swim, although it is slower than crawling or gliding. There are two different proposed mechanisms for amoeboid swimming. In the first, the cell extends small pseudopods which then move down the slides of the cell, acting like paddles. In the second, the cell generates an internal flow cycle with the cytoplasm flowing backward along with the membrane edge and forward through the middle, generating a force on the membrane which moves the cell forward.